Hello everyone, I am Dr. Tejashree Sardeshpande. I did PhD from Pune University and later postdoc from Bhabha Atomic Research Center, Mumbai. As seen in the earlier modules, flowering induction is governed by a number of external and internal factors which controls expression of flowering regulators FT and SOC1. Expression of these regulators bring about the conversion of the shoot apical meristem into an inflorescence meristem and further regulates downstream processes to specify the floral meristems on the flanks of the inflorescence meristem. We shall see how the inflorescence and flowers develop in the model plant Arabidopsis. The learning objectives of this module include: first, to understand how the floral meristem is formed; second, how the floral organ primordia specify the identity of the different whorls and determine floral organs; and lastly, we shall see some of the models that explain the flower development. Let us start then. As seen in the earlier module, in Arabidopsis, secondary inflorescences arise as branches on the primary inflorescence which retain the indeterminate nature of the primary inflorescence. After forming several branches, the primary inflorescence meristem begins to produce determinate floral meristems which develop into flowers. Floral meristem identity genes specify the floral meristems on the flanks of the inflorescence meristem. We have seen that the flowering locus T or FT and suppressor of constans or SOC1 are the flowering regulators which converts the shoot apical meristem into an inflorescence meristem in response to various flowering induction pathways. Another mobile protein called the terminal flower 1 TFL1 is a flowering inhibitor. TFL1 and FT share highly conserved amino acid sequences but have opposite functions. FT interacts with FD in the shoot apical meristem and activates the floral meristem identity genes, whereas TFL1-FD suppresses these genes. FT mutant increase the indeterminacy of the inflorescence due to lack of induction of the floral meristem genes, while TFL1 mutants show early determinacy, which leads to the formation of terminal flower. Together, FT and TFL1 play a role in fine-tuning the expression of floral meristem genes such that the floral meristems are only expressed on the flanks of the inflorescence meristem, while their expression is repressed in the inflorescence axis by TFL1, thus maintaining its indeterminacy. FT and SOC1 are involved in the specification of the floral meristem on the flanks of the inflorescence axis. The genes specifying floral meristems include a plant-specific helix-turn helix transcription factor LEAFY and a Mites-Box transcription factor a petala-1. SOC1 binds to the LEAFY promoter and activates its expression, while AP1 expression is activated by LEAFY along with FT-FD complex. Both LEAFY and AP1 activate genes involved in floral meristem formation and repress the flowering induction genes, thus maintaining the fate of the floral meristem. This cascade of regulation controls the precise timing of early events in the establishment of the floral meristems. Once LEAFY and AP1 are expressed, the environmental factors are no longer involved in the development of flowers. LEAFY mutants show a partial reversion of flowers into inflorescence shoot-like structures. AP1 mutants express a partial inflorescence meristem phenotype where secondary floral meristems appear in the axils of sepals. The LEAFY AP1 mutants show a complete conversion of flowers into inflorescence shoots bearing leaves on the primary and secondary inflorescence axis. LEAFY and AP1 are also involved in the specification of floral organs. Leafy mutants show flowers consisting of sepals and a central carpel, while AP1 mutants show flowers that lack sepals and petals, which are replaced by carpels and stamens respectively. We shall see their role in specifying floral organ identity in the later part of the module. Let us now see how floral meristem is formed. The stem cells in the inflorescence meristems are maintained by the homeodomain proteins Wuschel and clavata in the similar ligand receptor fashion as seen in maintaining stem cells in shoot apical meristem. The negative feedback regulatory loop of clavata and Wuschel maintains the stem cell pool constant during initial floral developmental stages. The clavata-Wuschel loop regulates the expression of LEAFY and apetala-1 in the flanks of the inflorescence meristems, which specifies floral meristems. In the floral meristems, Madsbox transcription factor A-gammas is induced by Wuschel and LEAFY at the center of the floral meristem. A-gammas directly represses Wuschel by forming a complex with a polycomb repressor complex family protein terminal flower 2. The stem cell activity in the center of the floral meristem is arrested and the zone of LEAFY and AP1 expression spans the entire floral meristem to specify the floral organ primordia, of which sepals are formed first. The A-gammas mutant display a flower-in-flower phenotype due to the maintenance of stem cells at the center of the floral meristem. Besides its role in regulating floral stem cell activity, A-gammas also plays a role in formation of reproductive organs which differentiate from the floral meristem. LEAFY is one of the first genes expressed in the floral meristem and plays a role in activating the transcription of specific floral organ identity genes in the distinct domains of the floral meristem. LEAFY activates floral organ identity genes by directly binding to their regulatory regions to form a distinct complex. The main floral organ identity genes are apetala-1 AP1, apetala-2 AP2, apetala-3 AP3, pistillata, and A-gammas. AP2 is upregulated by LEAFY alone, and AP3 gene is activated by a complex formed by LEAFY and another protein unusual floral organs UFO. A-gammas is activated by LEAFY along with Wuschel, and then A-gammas inhibits Wuschel to form the determinate flower. Let us now see how floral primordia develop into the mature flower. In Arabidopsis, flowers are attached to the receptacle and are made up of 4 whorls. First whorl is made up of 4 free sepals known as calyx. The second whorl, corolla, is made up of 4 free petals whose positions are alternate to that of sepals. The third whorl is made up of 4 long medial stamens and 2 short lateral stamens, and the fourth whorl consists of 2 fused carpels forming a superior gynoecium. The floral organ primordia develop from a small number of floral organ founder cells in the L1 layer of the floral meristem. The founder cells divide and expand radially to generate a concentric group of cells from which all floral tissues are derived. The 4 floral whorls are specified by the expression of distinct genes which impart identity to the floral organs in each whorl. Between the meristem and the newly formed organ primordia, a boundary is drawn by a specialized cells showing a lack of growth which separates meristematic activity from determinate organ growth. Similarly, the organ-organ boundaries are established where floral organ identity gene expression does not occur, allowing morphogenesis to proceed independently in each whorl to produce discrete organ types. Genes that defines and maintains sharp boundaries between the expression of other genes are called cadastral genes. For example, a zinc finger repressor protein called Superman or SUP defines a boundary between third and fourth whorl. In soup mutants, the third whorl extends into the fourth whorl to give rise to flowers with stamens in both third and fourth whorl. The fourth whorl does show carpel development, but the carpels are partially unfused. Let us now learn some more things about the floral organ identity genes. These genes are the transcription factors that regulate the development of specific floral organs. They are homeotic genes, which means that mutations in any of these genes lead to replacement of the original organ with another organ instead of inhibiting the development of the mutated organ. Mutants for the floral organ identity genes show replacement of the organs in the floral whorls. We have already seen that AP1 mutants flower show the absence of sepals and petals, which are replaced by carpels and stamens respectively. The AP2 mutants also show carpel, stamen, carpel respectively in the four whorls. The AP3 and pistillata mutants show sepal, carpel configuration, and agamous mutants show sepal, petal configuration, which repeats several times to form a flower within flower phenotype. Besides these mutants, it was seen that another group of genes called sepalata are also involved in floral organ identity. Triple mutants of the sepalata genes SEP1, 2 and 3 show flowers that consist only of sepals. The SEP4 single mutant displays a phenotype similar to that of wild-type plants, but SEP1, SEP2, SEP3, SEP4 quadruple mutants show the conversion of all floral organs into leaf-like organs, indicating the involvement of all four sepalata genes in the development of the floral organs. It also shows that all floral organs form leaf-like organs in the absence of developmental specification, thus supporting the fact that flower organs are modified leaves. Coming to the flower development model, it is known that flower organ identity mutants show development of two adjacent whorls except sepalata. The mutants are classified as Class A mutants, which affects sepals and petals, Class B mutants, which affects petals and stamens, and the Class C mutants, which affects stamens and carpels. This layout was represented as a model called the ABC model of flower development. The Class A genes are expressed in the outer two whorls and include AP1 and AP2, which are required for the formation of sepals and petals. The Class B genes are expressed in the second and third whorl and include AP3 and pistillata, which play a role in petal and stamen development. The Class 3 gene is agamous, which is expressed in the third and fourth whorl and is required for the development of stamens and carpel. In addition, the A and C function domains seem to mutually repress each other in their respective expression domains. In Class A mutants, the C function domain expands into the outer whorls, causing the expression of agamous in the first two whorls. In C class mutants, the A function expands to the inner whorls, which leads to the expression of AP2 in the inner two whorls. AP2 expression is seen in all four whorls initially and then becomes restricted to the first and second whorls by the post-transcriptional regulation of the AP2 mRNA in the C function domain. MicroRNA 172 suppress AP2 gene expression in the inner two whorls. Later, the sepalata genes were discovered, which are expressed in all four whorls. These genes do not provide any organ identity specification on their own, but are important for the expression of organ identity imparted by the A, B, and C class genes. The sepalata genes are called the E class genes. SEP1, 2, 3, 4 quadruple mutants show the conversion of all floral organs into leaf-like organs, indicating the involvement of all four sepalata genes in the development of the floral organs. This led to a revision of ABC model to include the E-class sepalata genes. This model is called the ABCE model. The ABCE model enables prediction of double and triple mutants of the floral organ identity genes. The AP2 pistillata double mutants show a loss of A and B class gene functions, which leads to the formation of carpels in all whorls, while the AP3 ergamous mutants show the loss of B and C class gene functions and develop sepals in all whorls. The loss of A and C class gene functions, as seen in a petala-2 ergamous double mutants, leads to the formation of carpeloid leaves in the first and fourth whorl and the formation of stamenoid petals in the second and third whorls. The triple mutant AP2 pistillata ergamous shows the development of carpeloid leaves in all four whorls. The formation of carpeloid leaves instead of leaves was due to the expression of another ergamous related genes like shatterproof SHP1 and 2 in the ergamous mutants. In addition, the ergamous mutants also show repetition of the respective pattern, indicating indeterminacy of the flower. Hence, the ABC model accounts for the phenotype seen in the double and triple mutants of the floral organ identity genes to a large extent. Beside mutant studies, ectopic expression of the floral organ identity genes also supports the ABC model. Use of the constitutively expressing cauliflower mosaic virus 35S promoter to drive the expression of floral organ identity genes leads to their expression in domains where they would have not expressed normally. In other words, the floral organ identity genes are ectopically expressed. The ectopic expression of the AP3 gene, which is a B function gene, gives rise to a phenotype that shows two outer whorls of the petals and two inner whorls of stamens. Since a petala-3 is expressed in all four whorls instead of being restricted to the second and third whorls. Similarly, the ectopic expression of ergamous gives rise to a phenotype of carpel, stamen, stamen and carpel, since ergamous is expressed in all four whorls instead of being restricted to the only inner two whorls. Constitutive expression of the floral organ identity genes, however, did not bring about the conversion of leaves into floral organs. When multiple floral organ identity genes like AP1, Sepalata2, Pistillata and AP3 were simultaneously expressed using 35S promoter in the same transgenic plant, it showed petals in place of rosette leaves. This is due to the presence of not only the A-B class genes functions but also the E class gene function. Without the Sepalata gene, the transgenic plant did not form petals. Hence, A, B and E class genes are required for specifying petals as already inferred from mutant studies. What about the D class genes? Why is this class is missing in the ABCE model? The D class genes define ovule identity and mutants of this class transform ovules into carpeloid structures. One D class gene in Arabidopsis is seed stick STK. The STK protein interacts with C class protein ergamous and E class protein sepalata to define ovule identity. However, since the D class genes are concerned with ovule development and not the development of the flower, they have been omitted in the ABCE model. In the ABCDE model, the ovule is considered as the separate organ, and since the ovule evolves earlier than that of the flowers in the seed plants, which includes gymnosperms, the D class genes are included in the model, where they along with C and E class genes define ovule identity. Another model of flower development is the bracketed ABC model, where the bracketed A is the floral identity function acquired at early stages of floral meristem development and necessary for all floral organ identities. In this model, the bracketed A includes the AP1, AP2 and Sepalata genes and has several roles. They are expressed before the B and C class genes and acts to establish the floral meristem identity and facilitates the production of the ground state of floral organs, namely the sepals. The bracketed A function later activates and regulates the B and C class gene functions. The interactions between the organ identity genes described by mutant analysis of the A, B, C and E class genes was later shown to arise from a protein-protein interactions in which the proteins coded by these genes were seen to form different tetrameric complexes. The Matchbox proteins are of two types. Type 1 match domain proteins only have their DNA binding match domain in common. The floral organ identity genes belong to the type 2 match domain proteins which have 4 domains in common. The match domain, I or intervening domain, keratin-like or K domain and a C-terminal domain. Hence, these transcription factors are also known as MIKC type of transcription factors. The Matchbox domain binds to DNA at the cysteine arginine glycine motif having the consensus sequence CC86GG. The I domain is involved in the formation of protein dimers, the K domain mediates protein interaction, and the C terminal is associated with transcription factor activation and the formation of higher order protein complexes. The quartlet model explains how the tetrameric match transcription factors bind to DNA to activate genes important in development of the floral organ in the specific whorls. Hence, AP1, CAL and sepalata interactions direct sepal development. Petals are specified by AP1 and sepalata together with AP3 and pistillata. Stamens are specified by AP3 pistillata combined with sepalata and agamous, while carpels are specified by agamous interactions with sepalata. Two dimers of match domain protein bind to adjacent cysteine arginine glycine boxes to form the tetrameric complexes, which leads to bending of the DNA between the two cysteine arginine glycine boxes. AP2, which is not a Matchbox protein, is not a part of tetrameric complexes. AP2 containing complexes act as a special regulators of gene expression during floral patterning along with the tetrameric complexes. Assembly of the MIKC tetramers creates new interfaces and recruits other proteins such as cofactors or epigenetic regulators to form multimeric complexes. Cofactors bind to cis regulatory elements in the vicinity of cysteine arginine glycine boxes and interact with the floral organ identity proteins. In addition, the transcriptional machinery recruits epigenetic regulators which can alter chromatin structure and accessibility by making post-translational modifications at histones. The MIKC tetramers have largely overlapping target genes and acts as transcriptional repressors as well as activators by interacting with different cofactors to regulate target gene activity. However, not all floral diversity originates from modification of floral Matchbox genes or their downstream targets. For example, changes in floral ground plan such as number and position of floral organ primordia appears largely independent of the MIKC gene activity. Sex determination mechanisms relies on changes in B-class gene activity. Still, much remains to be understood about the regulatory gene networks that bring about the expression of downstream genes. A large number of putative candidates have been identified using bioinformatic tools which are involved in determining the distinct morphologies and sizes of different types of floral organs. This awaits confirmation using experimental studies. To conclude, in this module, we have seen that flowering is a multi-step process. The first step decides the time of flowering, and this step is regulated by various external and internal factors. This step culminates in the conversion of vegetative shoot apical meristem into an inflorescence meristem which bears floral meristems on its flanks. Once floral meristems are specified, The plant goes through the flowering process without any further influences from the environmental factors. The floral meristems are determinate and gives rise to the floral organ primordia, which differentiates to form the four floral whorls. The earliest flowers were simple, just collection of carpels and stamens with no perianth. In fact, the perianth was often just a continuation of the leafy axis and not distinguishable into sepals and petals. Such flowers had numerous stamens and carpels which were free. The earliest flower, which is thought to have evolved about 140 million years ago, probably resembled the magnolia flower with multiple undifferentiated perianth consisting of tepals arranged in concentric rings. At its centers were multiple rows of sexual organs including stamens and carpels. In the next module, we shall study the development of the reproductive organs in flowers, namely the stamens and carpels, which play an important role in sexual reproduction.